Welcome again. So today now we will be discussing to our next chapter, that is the chapter 8, in which we will talk about the cell mediated immunity. So the immunity that will be used to kill the cell. So the cell will be of the foreign body but also of your own cell as well. That cell will be actually the altered cell, the damaged cell. So when you have any infection intracellularly inside the cell, suppose the virus, which are intracellular organism, or say some of the intracellular bacteria, they live inside the cell. We don't have any mechanism like antibodies that doesn't go inside the cell and kill that pathogen, either it is a bacteria or virus. So for killing the intracellular pathogen, we need to kill the whole cell, and the process by which the whole cell, our own cell, is killed is due to cell-mediated immunity. We know our immunity has been trained initially in such a way that it cannot recognize the cell peptide and kill our own cell, because in normal situation, if they will target their own cell, then we will develop the disease called autoimmunity. So we have not such mechanism to kill our own cell, despite that our own cell is damaged, diseased or altered. So we need to kill the cell, but only the infected one, only the damaged one, only the dying one. We cannot kill our normal cell. If we kill our normal cell, then that will develop the immune diseases like autoimmune diseases. But those cells which have been infected with the virus and bacteria, that need to be killed, because there are dying cell, that is the host, that is the food for the growing intracellular pathogen, and we need to eliminate from that, including the inside pathogen, the cell, including the inside pathogen from the body, and that is due by the cell mediated immunity. So let's revise. Let's see, this is the one of the diagram that was usually found in the old Kaplan microbiology book. This is from the, I think, 2013. So this is a 2013 USMLE microbiology book of Kaplan. So from there you can see this is the antigen that will be processed by this antigen presenting cell, and this antigen presenting cell will process this antigen by this MSC class 2 protein and then present it to the naive T cell. This naive T cell will check by the TCR, then check with the CD4, because this is processed by MSC class 2. Now if once they get activated, then they will be proliferated, either depending upon the signal, it is intracellularly or extracellularly. If the macrophage release the interleukin 12, then that will activate the Th1 arm, or it will, if they will activate, if the intracellular, there is the extracellular pathogen, then they will release the cytokine that will activate usually this antibody arm, that is the Th2 arm. So it's depend totally upon the protein nature and the nature of the pathogen. If it is intracellular, they will activate this Th1 arm. If it is extracellular, that will activate the Th2 arm. And we know in Th2 arm what will happen, this will form the release this cytokines like interleukin 4, 5, 6, 13 and TGF-beta. This all will help in the conversion of this B cell into the plasma cell and secretion of the antibody. This antibody will have the different function. We have discussed about the humoral immunity, where we have talked about this antibody can neutralize the pathogen, then can activate the complement from the MAC complex and kill the pathogen, and also helps in the opsonization of the pathogen and eliminating the infection. So that was the function of your Th2 arm, which is the humoral immunity. And they also release this interleukin 10 that actually inhibit the Th1 arm. So you have to understood, we have discussed this, that one arm is activated. If the antibody's arm is activated, it will actually inhibit the another arm, that is the cell mediated arm. So both arms cannot be acting at simultaneously. Either cell mediated immunity will be activated, or antibody dependent immunity or humoral immunity will be activated. But at a time, both cannot be activated, because they release the cytokines that have the capacity, that have the function to inhibit the other arm. So at once only one arm will be activated, and that will be more powerful, that you have to understand. So if we have activated the Th2 arm, then that would release this process the antibody secretion, but they will also release the cytokine called interleukin 10, and this interleukin 10 actually what will do, this will inhibit this your Th1 arm, and then Th1 arm will be not be activated. If you have given intracellular pathogen that will be processed and presented mainly by the MSC class 1, that will goes to the TH helper T cell or cytotoxic T cell, and cytotoxic T cell that will release this if TH1 arm is activated, then this will release the interferon gamma, and this interferon gamma will actually inhibit the TH2 arm. So at a time only one arm will be activated. So since we are talking about that is cell mediated immunity, then we are talking about the TH1 arm, and TH1 arm will produce the interleukin, that is the interferon gamma, and this interferon gamma, once the TH1 will be activated, actually this depends upon the interleukin 12 that is will secreted by the macrophage. If the macrophage will unable to kill the intracellular pathogen, then they will release the interleukin 12 that will activate the TH1 arm, and this TH1 arm will actually release the interferon gamma, and this interferon gamma will inhibit the TH2 arm, and also this interferon gamma including the interleukin 2 and TNF beta, they will activate the certain cell that will kill the whole cell including the pathogen that is present inside that cell, that is the intracellular pathogen. Okay, so what are the cell that will be activated? They will activate the cytotoxic T cell. What will do? This cytotoxic T cell will kill the virus infected cell and tumor cell. This will activate this NK cell. This NK cell function is also to get activated and kill the virus infected cell as well as the tumor cell, and they can also kill the whole cell by the process called ADCC. ADCC is antibody dependent cellular cytotoxicity, and in this process what will happen? This antibody has one site where they bind through the disease cell. So this antibody will bind to the disease cell, and their FC receptor has the binding capacity to the other cell like neutrophil, macrophages, and then they will kill this altered cell or damaged cell. So this is called antibody dependent. This the mechanism that is involved, the intermediate product or intermediate bridging that is involved with antibody, so it is called antibody dependent cellular cytotoxicity, and this is ADCC. So what is the function of your TH1 arm? They will activate actually effector cell of the cell mediated cytotoxic. They will activate the macrophage, and this macrophage have the capacity to kill the pathogen very fast. Mainly in case of tuberculosis, the tuberculosis organism, they will activate the TH1 arm, and if the TH1 is activated, the activated macrophage becomes now so powerful that they can kill all the bacteria. So when tuberculosis is facultative intracellular pathogen, so when they are inside the cell, they are not able to be killed, then they activate the macrophage. This activated macrophage has increased capacity to clear the mycobacterial infection. So when TH1 arm is activated, they will release this interferon gamma, and this interferon gamma will activate the macrophage activation that will becomes more powerful. They also activate the CD8 NK cell and antibody dependent cellular cytotoxicity. So the main concept, what I am going to explain to you, what is the take home message is that when we are talking about the cell mediated immunity, means the whole cell is going to killed by our immune system. Now that whole cell includes the whole cell of the virus, whole cell of the bacteria. Obviously the bacteria and virus inside our cell will be killed, but all other normal thing will also be killed, because our cell is dying disease, but it will not damage to the normal cell. That we have to understand. Okay, so this is not about the killing one bacteria or one pathogen. This is about killing the disease, dying, damaged our cell, that you have to understand. Okay, so if you have understood this, then let's come back to this Kaplan microbiology. This is in which we are talking about this chapter 8, that is the cell mediated immunity. This is our 2018 or recent book where we will talk about this cell mediated immunity, and cell mediated immunity has evolved into battle in two different types of pathogen, that is the facultative intracellular pathogen and obligate intracellular pathogen, which adapt to live inside the phagocytic cell that are designed to kill them, where obligate intracellular pathogen which can't replicate outside of the host cell. Cell mediated immunity is detected by the TH1 response. We have discussed about this. This TH1 response is called the cell mediated immunity, primarily via the macrophage and CD8 cell, while the TH1 response is generated towards the eliminative intracellular pathogen. The main thing is to eliminate the intracellular pathogen. TH cell in general direct all aspect of the immune system. Naive cell that will orchestrate, that will manage both in TH1 and TH2 arm, that is the TH0. But when you talk about the TH1, that will be intracellular pathogen elimination. The primary mechanism by which T cell direct all aspect of the immunity is called by the secretion of the cytokine. We know these chemicals that are called cytokines. NK cell also have a role in this type of immunity. They were covered initially. The macrophages, we know the macrophages, Th1 response activated macrophage and B cell via the cytokine interferon gamma activate the classical M1 macrophage to eradicate intracellular pathogen and induce B cell to class switch to produce opsonization of immunoglobulin G antibody that can assist the macrophage with phagocytosis. So we have initially discussed about this, that interferon gamma, that is INF gamma, that can also cause class switching of the immunoglobulin G. So initially from the class switching of the immunoglobulin G is usually depend on the interferon gamma. If you talk about the interleukin 5, that is responsible for class switching of the immunoglobulin A, and if you talk about the interleukin 4, that was responsible for the class switching of the immunoglobulin E. So we have discussed about a lot of things. Okay, so we have discussed about how this class switching occurs in the humoral immunity. Now we are discussing about the macrophages, and macrophages actually interferon gamma cause M1 macrophage activated and also cause class switching of the immunoglobulin G. So this you can see over here, this is the antigen presenting cell, that naive T cell that activate the Th1 arm which release the interferon gamma. This interferon gamma cause macrophage to activation, that is the M1 activation which cause the classical macrophage activation and enhance the microbial killing. They also this acts on the B cell and cause the class switching of this immunoglobulin G that acts on the that acts as the opsonization and phagocytosis of the cell. So this is one of the way of cell mediated immunity as well, related to the macrophage and antibody. Okay, macrophage T cell interaction, binding of the TCR of the naive T cell to the MSC class 2 peptide complex of the macrophage provide the first signal to the T cell to become it activation. We know about this, talked about this T cell activation. There is the co-stimulatory signal, then this get activated, and then there will be the clonal expansion, and then T cell becomes into the effector cell and memory cell. The effector cell leave the secondary lymphoid tissue, enter into the circulation and travel to the site of infection, which we have talked about. So this is all about the site of infection. There will be the draining of the antigen presenting cell will be drained, that will process and present that antigen to the local lymph node, regional lymph node, where this will be in the paracortex region. There are the T cell. This T cell will be activated, then they will actually convert into they will activate the B cell, and B cell will convert into the plasma cell and form the antibody. This is about the cell site of infection. This antibody and effector B cell will go, and then they will deal with the infection. This was with this we have discussed already in the humoral immunity. Okay, and this was the tissue infection is drilled by this lymphatic vessel, where if it is in the blood infection or microbials is present, that will deal by the spleen, and this is what we have discussed in the humoral immunity. But one more thing you have to understood about this is cytotoxic T lymphocyte, which is the part of your cell mediated immunity. Cytotoxic T cell, or another name is CD8. This CD8 recognize the cell will be ultimately killed by interaction between the TCR and MSC class 1. So we have discussed about this MSC class 1 and obviously T cell receptor. This is rule of 8. If the cell question is performing normal function and therefore producing self peptides, there should be no CD8 T cell that have a complementary TCR structure. So if there is a normal MSC class 1 is present in each and every cell of our body, except including platelet, except RBC. So they are present everywhere, and every cell, actually all antigen presenting cell has both MSC1 and MSC class 2 protein. MSC class 2 is only present on the APC, antigen presenting cell, and that is macrophage, dendritic cell and B cell. All other cell, but this 3 cell has also MSC class 1, including all other cells that have the MSC class 1 except RBC. Okay, and if the MSC class 1 is adequate, is normal, then CD8 will not bind. This will be only bind when there will be process and present the intracellular pathogen, which we have discussed about it, and then will brought to the MSC class 1, means the cell is dying. Okay, and then only the CD8 cell will be activated. If the cell is infected with the intracellular pathogen or is expressing new antigen reflecting on the tumor transformation, some small portion of those CD8 cells generated from the thymus should be capable of binding this TCR to this MSC class 1 non-self peptide complex, so which we have discussed over here. Unfortunately, because of the extreme polymorphism of the HLA MSC system in the human, when tissue are transplanted between non-identical individual, cells of the transplant are often targeted by CD8 as abnormal. So when we are talking about this transplant, so what will happen, except the bone marrow transplant, in other transplant patient, if you transplant certain substances, that cell have the MSC class 1, but that is of the another portion. So for me, that becomes, for my CD8 cell, that become a target, and this CD8 cell will go and attack that cell, and because of that there will be the rejection of the transplant. So this is all because of your cell mediated immunity. You know, if any foreign thing is inside your body, obviously your immune system is trying to clear that. And how it can be cleared? Because you know, if you transplant any identical person, you will transplant the MSC class 1 of my body, and any person will be totally different. So now this MSC class 1 will be foreign, and when this CD8 cell will found this different MSC class 1, then that will attack that cell, and that will cause rejection of the transplant. So this is the main pathogenesis of your rejection of your transplant. So CD8 cell kill the target cell by delivering the toxic granule content that induce the apoptosis of the cell, to which they attach. This process occurs in 4 ways: attachment to the target cell mediated by the TCR CD8 cell, okay, and LF1 integrin. Now activation occurs, rearrangement of the consanguine granules against the attached target site, then exocytosis of the granules, perforin and granzyme, and detachment from the target cell. So all, if you remember, NK cell or say your this CD8 cell, this both will act and cause damage or cause killing of the cell by the 2 process. One is they will attach to the cell, then they will release this enzyme that is called perforin and granzyme. So the granzyme enters through the hole, through the perforin holes, and activation of the caspases. So what happened when the cells get attached, how the cell as a whole cell will be killed, and that will be killed by one of the mechanism called the enzyme called perforin and granzyme. This perforin will first be released, that cause pores in our cell, and once there is a pore, then granzyme will be entered into the cell, and once granzyme will be entered inside the cell, that will activate the caspase of death domain. So if the death domain, they will activate the caspases, and this caspases will activate the death domain, and then once the death domain is activated, the cell dies. So we have a switch inside our cell that is called death domain. If you on that switch, then our cell will die. Our cell will undergo apoptosis, and it will die. Similarly, this is the mechanism of CD8 and NK cell. If you talk about another mechanism called the fast ligand, so fast on the target cell interacts with the fast on the CD8 cell. So this is another mechanism where fast bind with the fast ligand and then cause the apoptosis of the cell. So in this way, cells is killed by cell mediated immunity. So there are 2 mechanisms, perforin and granzyme. Perforin this will be the enzyme secreted that will create the pores inside the cell, and the granzyme will be entered that will activate the caspases and activate the death domain, and the cell is killed. Another is fast ligand, fast and fast L. So fast will bind with the fast ligand, and then there will be the activation of the cell. The apoptosis of the target cell will be done, and the cell will be killed. We have discussed about this NK cell. NK cell is also the same mechanism. They will also release by this perforin and granzyme, and only to those cells which have the expression of the inhibited by normal expression of the MSC class 1 via HLA-E. So if there is normal MSC class 1, this NK cell will be not going to kill. But if there is any abnormal, so what will happen if there is intracellular pathogen infection?